Continuing on the topic of species diversity, we're going to be talking today about species turnover. As we've done in the last couple of sessions, we're going to continue talking about elements of the composition of species in a community. We've talked first about species richness, that is the number of different kinds of species there are, and then we talked about how diversity is affected by both richness as well as relative abundance. Now, if you will recall, we had touched upon the idea of alpha, beta and gamma diversity. Alpha is essentially the diversity within a unit area. Beta is the difference in diversity or differentiation diversity between two areas, and this can be examined over multiple scales within samples, between samples, within habitat, between habitat, and at landscape scales and at the biogeographic province scale. And these have been designated as alpha, beta, gamma, delta, epsilon and so on. But as mentioned, the two broad elements of these are alpha, which are within a particular unit area, and when we compare another similar unit area, the difference between this becomes beta, and this is overall gamma. Now, I could also designate this as alpha and compare it to some other equally large area and call the difference between these beta. So even though there are sort of sample, habitat, landscape and province level distinctions are made in the literature, there are differences in the scales to which it's applied, and it's useful for us to keep in mind that alpha, beta and gamma in some sense give us the three measures that are of interest to us and can be applied at depending on the ecological questions that we have. Now, these comparisons could be, for example, between tropical and temperate forests, but that's the difference in composition between tropical and temperate forests is vast. There are probably very few or perhaps not even any species that overlap between the two, except perhaps in places where tropical and temperate zones overlap. But more often we're interested in what happens when you move from one forest to another across peninsular India or across the Western Ghats or across the Himalayas, and so you go from one forest type of one forest area to the other, and you're like, oh, how much have tree species changed? I'm still in an evergreen forest, but are the forests of southern Western Ghats the same as the forests of the northern Western Ghats? Are the trees within the rainforests in the Anamalays the same or different from those in Mudumalai, and what underlies these patterns? Another sort of nice model to look at are these shola forests that are found in the upper reaches of the southern Western Ghats. These montane ecosystems comprise these rolling grasslands with little patches of forest, and one can just look at this image and intuitively wonder about what the difference between tree species composition or any faunal group composition might be between these. In order to understand this, we need to measure beta diversity, and as mentioned, this is essentially a way to quantify the differences in composition between two areas. As you can see, for example, from these two in area 1 and area 2, there are these species, this one, are common between the two, this guy as well, and the others are not. How do we go about measuring this difference? One of the most common ways to approach this is to examine the difference as as a relationship between alpha and gamma diversity. Now, you can also examine the differences in species composition between the two areas directly, and you can use the species area relationship and measure turnover relative to species accumulation with area. Now, we are going to examine the first two in some detail. One of the most commonly used measures of beta diversity was Whittaker's beta, which is S by alpha minus 1, S being the total number of species recorded in the system, and alpha being the average sample diversity, and with the assumptions that each sample is equal and diversity is equal to species richness. Now, essentially, if I have 3 areas where I've recorded 3 species here, 9 species here, and let's just say 6 species there, and some are obviously common, and so perhaps the total number of species across all of these is let's just say 12, right? So S is equal to 12, and alpha, which is the average of these, is 6, right? So my Whittaker's beta is 12 by 6 minus 1. Now, the maximum value that I could get here if beta was really high is if all of those species were different, in which case my average species, the number of species that I would have over here is 15, and my value of beta would be correspondingly higher. Similar measures of beta diversity have been proposed. Cody uses a change in composition along a gradient. Routledge partitioned beta into alpha and beta components, and where in this particular form R is the number of species pairs with overlapping distributions. But another element of beta that was important to consider and compute was that beta can come about from 2 different processes. It can come about from nestedness and it can come about from replacement. So, for example, if I have 5 species and I have 3 areas, 1, 2 and 3. Now, let's say that area 1 has A, B, C and D. Now, if area 2 has just A and B, 2 is said to be nested within 1. So, the species in area 2 are a nested subset of the species in area 1. Now, on the other hand, I can have beta diversity come about as follows. Area 3 has species E and F, which area 1 does not, but doesn't have the species that area 1 does. And this comparison of 1 and 3 actually, or in fact of 2 and 3, is that some species have been replaced by others. Now, both nestedness and replacement both contribute to beta diversity, and in many instances when we are comparing communities, there is both nestedness and replacement. So one of the approaches to understanding beta diversity is also to partition where beta is coming from. Is it coming from nestedness or replacement? Now, again, Wilson and Schmida provide an evaluation of measures of beta diversity. They point to a number of factors such as the number of community changes, whether it's additive, its independence from alpha, its independence from sampling, and they came up with an index which fulfilled the largest number of these criteria. Similar but slightly different approach is the idea of complementarity, while beta diversity looks at the differences between areas, the similarity measures have been used for understanding how similar two areas are. And the idea of complementarity, though it's the opposite of similarity, is that we want to maximize the number of species that from a certain number of areas that we might be prioritizing for conservation. Now, the reason we do this is that we might have a number of areas of very, very high diversity, and a measure of an approach to conservation planning that sort of takes into account the high diversity areas may actually conserve 5 patches, but all 5 patches that are very similar. Now, there might be a 6th patch that's has lower diversity but is substantially different from the others. So it actually then helps to maximize the total number of species, or the gamma diversity of the areas that I'm setting aside for conservation, by looking at what patches are complementary to each other in terms of the species they have. Therefore, high beta diversity not from nestedness, but high beta diversity from replacement. The measure is of complementarity is given by this equation, where A is the number of species in both quadrats, B the number of species in quadrat 1 or area 1, and C the number of species in quadrat or area 2. Widely use a number of similarity coefficients in this when addressing these questions, and this relates closely to the underlying ecological questions about what drives these patterns in nature. Some of the common ones are Jacquard's index and Sorensen's. In both of these, J is the number of species found at both sites, A is the number of species in site A, and B is the number of species in site B. Now, these similarity coefficients only take into account species presence and absence, but there are quantitative measures. Sorensen's has one which takes into account the total number of individuals at the 2 sites, and then which is incorporating the number of individuals in site A, site B, and the sum of the Jn, which is the sum of the lower of the 2 abundances for species found in both sites. A widely used quantitative measure of similarity is the Morishita-Horn index, which has been found not to be sensitive to species richness and sample size. I'm not going to go into the details of the formula here, but again, it takes into account the number of individuals at both sites, and also the number of individuals of particular species across the 2 sites. Now, these are the measures that help us understand the degree to which 2 areas might be different from each other in terms of the species composition. One of the fundamental reasons that we're interested in this is either in terms of ecological gradients. So as you go from the southern to the northern western Ghats, as you go from lower to higher elevations in the Himalayas, as you go from the south to north in the Andes, why does species composition change? And broadly, these have been categorized as dispersal and niche theories of species composition. And dispersal assembly argues that community composition is determined by the dispersal ability of species and by chance. So different taxa might have different dispersal abilities, and there's a certain amount of stochasticity in where propagules of either fauna or flora reach. On the other hand, niche assembly argues that community composition is determined primarily by environment, that once a propagule has reached a particular location, there's an environmental filter, there are abiotic factors that enable species to survive there. So if I if I compare the southern and the northern Western Ghats, there are differences in temperature, there might be differences in rainfall, in seasonality, and these differences drive actually the difference in composition. This is what niche assembly would argue. In reality, there's greater and greater recognition that both dispersal and niche assembly play a role, and what we're really trying to quantify or infer is the relative roles of the two, but there's variation variation across scales, there's variation across types of habitats, and there's definitely variation across taxa, because obviously taxa that have greater dispersal ability will show stronger signals of dispersal assembly at particular scales than other taxa. Fairly recently, there's been an examination of what one might expect from niche and dispersal assembly hypotheses. What's plotted here is the immigration rate on the x-axis and species richness on the y. And as you can see, immigration rate makes no difference to species richness in the niche assembly hypothesis because dispersal doesn't play a role there. And in the dispersal assembly hypothesis, there's a linear monotonic increase with of species richness with immigration rate. Now, as I just mentioned, we're largely looking at systems where we know that both niche and dispersal assembly play a role. Lok and Chisholm argue point out that the classic unified hypothesis suggests that when there's dispersal assembly, there's an increase in the number of species richness, and once dispersal has saturated a particular area, niche assembly takes over, and immigration no longer influences species richness. They, however, propose that many habitats are already saturated, and in fact, in niche assembly, there's no the communities start out with niche assembly where immigration rate does not affect species richness at all, but dispersal assembly then actually oversaturates communities, and communities have more species than niches, and so they propose that niche and dispersal can work together, but somewhat differently from how the classic unified hypothesis frames it. Finally, coming to the ways in which we actually examine this, we've mentioned the idea of using these metrics to understand underlying ecological or evolutionary patterns. So let's go back to that shola grassland system and say, for example, that we sampled 10 sholas in this landscape, and let's take trees as a taxon again, because trees don't move, and we know that each of these that individual animals are not moving across these sholas in this fairly small spatial scale. Now, we've gone out and measured trees in all of these, and we have tree species composition for all of these different sholas. And ecologist colleague of mine and I argue, one of us is a dispersalist, and I, for example, I argue that sholas that are closer to each other are more likely to be similar in composition. This is derived from the idea of dispersal assembly, the trees dispersing or the seeds dispersing from one shola to the other reduces with greater distances, and therefore sholas that are geographically or spatially close should be more similar in composition. My ecologist colleague argues that actually dispersal doesn't play much of a role. They can all disperse fairly far, and it kind of depends on where the shola is located. Is it in a valley? Is it on a slope? Is it east facing? Is it west facing? What's the microclimate? What's the soil? And he argues that actually in this particular landscape, the specific environment determines what trees are and aren't found in each shola. At larger spatial scales, you can imagine that that difference in environment could be much greater. It could be actually differences in temperature, rainfall, and a whole range of other abiotic factors. So now, how do I go about answering this question? One of the ways is to look at them as pairs. So I have pairs of sholas, and for each pair, I can generate a matrix of similarity of species composition, right? That similarity matrix tells me how similar or dissimilar each shola is in composition from any other shola. So in this, I have 1 to n, of course, but let's just say there are 5 rows. So if I had measured 5 sholas, I would have 5C2, and therefore 10 pairs of similarities of species composition. But I can also similarly have 10 pairs, a matrix of geographical distance, which gives me the pairwise distance between each shola, and I can similarly calculate an environmental distance between the 2 sholas, right? Between any pair of sholas. So I have now 3 matrices, and we are normally, when we have a variable such as species composition, geographical distance, environmental distance, we might be able to, we might statistically run a regression analysis on it. Because these are matrices and those individual data points are not independent, each data point actually is derived from a pair. So all of the points in this row, there are 4 points here. The pairs of 1, 2, 1, 3, 1, 4, and 1, 5. All are based on a comparison with area 1, right? And so those points are not independent, and so we can't use conventional methods to analyze this. This approach could be used for the analysis that we're using. It could also be used to compare other kinds of matrices, such as morphological similarity with geographical and genetic distance, and so on. And what we do in this case is that we can conduct a statistical test of correlation between the matrices, and these matrices must obviously be of the same dimensions, which they will be, because we're basically taking these areas of these sholas and computing pairwise matrices of A similarity, species similarity, B geographical distance, and C environmental distance. And we can compute a particular correlation between each of those between each of the matrices for each pair. And since we can't use a conventional regression or correlation approach, we use permutation or randomization tests. More recently, there have also been multiple regression approaches to matrices which extend the Mantle test. There's also been criticisms of these, that they lack a framework for fully specifying null and alternative hypothesis, and that the tests can be flawed when there is spatial autocorrelation, which returns erroneously low p-values. To examine these questions, that we are still interested in these questions, we still want to know whether geographical distance or environment determines the similarity between 2 sholas in that landscape. More recent approach is variance partitioning, which is an extension which is based on redundancy analysis. That's an extension of the principal components analysis. It was developed over the last couple of decades for the kinds of questions that we've just asked, and essentially our data set now would comprise multiple response variables. The response variables being the species composition, or the presence or absence of species in each of our sites, and of environmental variables. And we can then partition the variance in that data set into distance. So how much of the variation in the species composition, the presence or absence of species, in our example in the sholas, is explained by distance or space, how much is explained by environment. But there is an additional component that environment itself tends to be spatially correlated, and so we can also tease out the component that is spatially determined environment. These different approaches are now getting us to answering those questions that we have about what's driving the differences in composition, and those predominant mechanisms being dispersal or distance, and niche or environment. We haven't actually spoken explicitly about biotic interactions influencing species richness. There are obviously biotic effects in terms of predation, competition, and other interactions. We haven't explicitly included those in these analyses yet, but obviously in order to fully understand a system, understand its richness or its diversity that incorporates abundance, or to understand turnover or beta diversity, we have to examine all of the elements that drive processes within these systems. Thank you. That's it for today, and we'll be back with a closer examination of relative abundance itself.